Hello, I'm Ross Pedersen, here to tell you about our recently published JCB paper entitled Endocytic Myosin-1 is a force-insensitive power-generating motor. This manuscript describes work that I did as a graduate student in David Drubin's lab at UC Berkeley, in collaboration with Aaron Snoberger, a postdoc in Mike Ostap's lab at UPenn. Together, we studied a myosin motor that is essential for clathrin-mediated endocytosis, and we found that this particular myosin is a relatively powerful motor. During clathrin-mediated endocytosis, actin assembly and recruitment of monomeric myosins, called type 1 myosins, occurs at the time of vesicle internalization. Actin is not required for every clathrin-mediated endocytosis event, but the extra force it provides is critical when membrane tension or turgor pressure oppose plasma membrane deformation, as they do in one of our favorite model organisms, the budding yeast Saccharomyces cerevisiae. We've long thought of clathrin-mediated endocytosis in this organism as being a process driven mainly by actin assembly-based forces. In this model, coat proteins at the tip of the membrane invagination bind to actin filaments, and new actin monomers are added near the plasma membrane, creating a driving force that separates the tip of the invagination from the base. But actin assembly isn't the whole story, because the type 1 myosins in this organism, myo3 and myo5, are also required for endocytic internalization. How myosin activity contributes to an actin assembly-based force generation mechanism is a long-standing mystery in this field. In general, proposed roles for these myosins fall into two categories. They could generate power to assist actin assembly in invaginating the membrane, or they could sense force and serve as anchors that prevent the actin network from assuming unproductive orientations. To distinguish between these two possibilities, we set out to study myo5 in vitro to determine how force-sensitive it is. We reason that if myo5 can readily overcome resistive forces, it would be ideally suited to generate power during endocytosis, while if it is acutely force-sensitive, it would be better suited to act as an actin anchor. We purified a truncated myo5 motor lever construct along with its light chain, and reconstituted myo5 motor activity by combining the two purified proteins with actin filaments and ATP. This in vitro motility assay, where fluorescently labeled actin filaments glide over a myo5 coated cover slip, illustrates how we could reconstitute myo5 motility on a surface. We could also reconstitute this reaction in solution, which allowed us to use stop flow spectrometry to measure rate and equilibrium constants of key steps of the myo5 ATPase cycle in the absence of force. Once we had measured the unloaded motor properties of myo5, we turned to single molecule experiments using an optical trap to measure how myo5 motor activity varies with resistive force. We used dual optical traps to create an actin dumbbell between two beads, then held the actin close to a third bead that was sparsely coated with purified myo5 and recorded binding and displacement events. Because we had already measured myo5 properties in bulk experiments, we could quantitatively test specific predictions, like how actin-myo5 attachment durations should shorten with increased nucleotide concentration, which you can see is borne out here, validating our approach. By challenging the myo5 power stroke with resistive force, we could also measure the force sensitivity of myo5 activity. Plotting the myo5 detachment rate as a function of interaction force reveals some degree of force sensitivity, with myo5 detachment slowing under resistive force. However, directly comparing the force sensitivity of myo5 with that of related motors puts this observed force sensitivity into context. Myo5 is considerably less force sensitive than other myosin-1s, and it's more comparable to cardiac myosin. Myo5's relative force insensitivity and fast working stroke mean that it generates significant power over a wide range of resistive forces, in contrast with other myosin-1s, which generate less power and are more easily frustrated. Based on these results, we conclude that myo5 is better suited to generate power during endocytosis. And overall, our work reveals how two cellular mechanisms that power movement, typically thought of separately, can additively contribute to a membrane-bending process. It'll be interesting in the future to untangle the relative contributions of each mode of power generation in this process and in others.